Today I want to say about the class 10th and biology subject. The topic is moles of leaf experiment. The CO2 is essential for photosynthesis. This is very important for public examinations for 10th class students. And what is the aim of this experiment? The aim of this experiment is to prove that CO2 is essential for photosynthesis. To prove that CO2 is essential for photosynthesis. And the apparatus, what are the apparatus we have to use for this experiment? Means actually a wide mouthed bottle. This is a wide mouthed bottle. And KOH solution. And potted plant. And also split cork, wax, and also the iodine solution. So these are all the apparatus we have to use for this experiment. And next, what's the procedure of this experiment? Means actually first we have to take one potted plant. For this experiment we have to take one potted plant. That potted plant should kept in dark condition nearly 2 or 3 days of time to avoid the formation of that carbohydrates. To avoid the formation of carbohydrates or glucose. So we have to keep this total potted plant nearly 2 or 3 days of time in dark condition. Then we have to take one wide mouthed bottle. This is wide mouthed bottle. We have to pour some quantity of KOH solution in the inner side of the wide mouthed bottle. Or we have to keep KOH pellets also. If pellets are KOH solution, whatever it may be, we have to keep inside. If pellets we keep there means there is no need of KOH solution. If our KOH solution we have to pour in the wide mouthed bottle. At the next, at the wide mouthed bottle's mouth region, at the mouth part of the wide mouthed bottle, we have to keep one splitted cork, and then we have to arrange like this total experiment for the completion of this experiment, and one of the leaf should be kept in the inner side half portion and outer side half portion through this split cork, through this splitted cork. There is a split in the cork region. That's why that through that splitted cork we have to keep half of the leaf in the inner side of the bottle and half of the leaf should be outside of the bottle. And here through that splitted cork we have to apply some grease to avoid the entry of any gases towards the inner side of the wide mouthed bottle. And total this apparatus should keep for sunlight nearly 3 to 4 hours of time. And then finally what will happen means after completion of 3 to 4 hours of time, we have to take this total leaf outside, and that leaf should dip in iodine solution. It should be dipped in iodine solution. So finally what we have to observe in this experiment means the half part of the leaf which is outside of the wide mouthed bottle only turns into bluish black color, which indicates the presence of starch or glucose, and whereas the half part of the leaf which is kept in the inner side of the wide mouthed bottle should not change that color. It is pale in color. It should not change. It should not change its color into bluish black because there is no possibility for photosynthesis. Why that possibility not happened here? Means actually the outside part of the leaf it receives sunlight. It receives sunlight or light. It receives sunlight and it receives carbon dioxide which is present in the outside. So CO2 is also that part of the leaf was observed. And for photosynthesis, what are the essential ones? We have to remember that is most important. Light, CO2 and chlorophyll, that is already present in leaf. That's already present in leaf. So there is no problem. So chlorophyll is already present in the leaf. CO2 is absorbed by the leaf from the atmosphere, and light is also it received. Light also it received. And chlorophyll, CO2 and light. But whereas the half part of the leaf, if we observe, CO2 is not observed by the inner side of the leaf. It also receives the sunlight because this wide mouthed bottle is very transparent bottle. So it also receives that sunlight, and the chlorophyll are also present. But the CO2 is not entered in the wide mouthed bottle. If any lesser amount of CO2 which is present in the inner side of the bottle is also absorbed by the KOH solution in the experiment. Actually, why this KOH solution we have to pour in the wide mouthed bottle means why that KOH solution will absorb the CO2 which is present in the inner side of the bottle. So that leaf does not receive any CO2. So finally that total leaf, if we dip in the iodine solution, only half part of the leaf which is outside, that only turns into bluish black, but whereas the half part of the leaf which is in the inside of the bottle should not change its color, so it is already pale in color. But so that is very essential here. So CO2, if it receives, that half part of the leaf receives, that can able to prepare the food material or starch or glucose, whatever it may be. But this half part of the leaf which is in the inner side of the leaf and the inner side of the wide mouthed bottle does not receive any CO2. That's why it does not able to prepare the food material. That means starch. It does not able to prepare the starch. That's why it should only in pale color. And what are the precautions we have to take in the experiment? Means the inner side of the leaf should not touch with the KOH solution. So if leaf touches with the KOH solution, what will happen? That leaf will absorb immediately that KOH solution. So that or if that leaf absorbs that KOH solution, means that total CO2 which is present in the inner side of the wide mouthed bottle should be there only. So that's why it and that's a leaf should not touch with the KOH solution. This precaution we have to take compulsory. Next, we have to apply some grease towards the split of the cork. Why? It should not give any entry of any gas towards the inner side of the wide mouthed bottle. So we have to take that precaution also. And what is the result? Finally, we have to get means actually CO2 is essential for photosynthesis. CO2 is essential for photosynthesis. Without CO2, it cannot be able to prepare the starch. So this experiment is very essential for 4 marks, and this experiment is also sometimes in public examinations they will ask what is the moles of leaf experiment. Discuss. They will ask something like that. Or sometimes they will ask, they will give only this diagram. What is the aim of this diagram? They will ask. So at that time what we have to write in the examination? So what is the aim of this experiment means? To prove that the CO2 is essential for photosynthesis. What are the apparatus we have to use to complete this experiment? They will ask such type of questions. What are the apparatus means? Here potted plant, rubber cork, wide mouthed bottle, KY solution, and finally iodine solution also. So those are the parts. And what are the observations we have to take in the experiment? Means leaf exposed to air becomes bluish black, but the inner side of the leaf does not changes into bluish black color. That observation we have to write. And what are the precautions we have to take for this experiment? Means the inner side of the leaf should not touch with the KY solution, and grease should apply at the region of splitting, which is kept in the wide mouthed bottle's mouth region. So to avoid the entry of any gas, that precautions we have to write. And what's the result? Means CO2 is essential for photosynthesis. So this experiment is very simple one, and if we write these points for 4 marks, we will get full marks. Sometimes they will ask aim, apparatus, precautions, observations, or result, whatever it may be. They will ask one by one questions by giving this total diagram. So we have to remember total all these points, and then we have to write very nicely. And if you like this class, please subscribe my channel. Thank you to all.